In this podcast, we will go over the procedures for using the paper planting wedge. First, we will talk about creating and printing out the paper wedges. Then we will go over putting the seeds on them for cold treatment, and finally we will describe the procedure for planting out an experiment using the seeds on the paper wedges. Let's begin. Before planting, our Arabidopsis seeds need to be cold treated. In a previous podcast, we introduced you to planting the seeds out in soil and then cold treating the whole tray. Here we will discuss a space-saving alternative, the planting wedge. These are labeled filter paper discs that seeds can be placed on top of and then put into the incubator. To make the discs, follow the template file provided. You can access it from the Google Groups page or from your lab's PI. The numbers that are printed on the discs are reference numbers, not names of lines. These reference numbers should be associated with specific lines in a database before you begin. To start, print out one template page on a regular 8.5 by 11 sheet of paper. Then, align a piece of 10 centimeter filter paper over each circular set of numbers and affix it with a small piece of Scotch tape. Make sure the tape is on the same side of the filter paper. For the rest of the discs, use a new piece of paper and tape filter paper on top of it following the one you just did. You should be able to see through the paper to align it. Now, feed all of the sheets with filter papers taped to them into the printer. Make sure that the taped side of the filter paper is going to be fed into the printer first so it does not jam. Press print. The numbers should now be printed on the filter paper. You may also want to use a pencil to write in the sulk lines on the filter paper as another check so that the reference numbers remain associated with the correct lines. Now you have to partially cut the 8 wedges in the disc. Leave them attached at the center, don't cut them apart. Place each cut disc into a Petri plate with a cover. Gather your supplies for placing seeds on top of the plate. You'll need a wash bottle with water, a paintbrush, a white piece of paper in case you drop seeds, and small beakers of water and ethanol that you will use to sterilize the paintbrush. Using a wash bottle, squirt just enough water onto the plate to wet the paper. Too much water will cause the seeds to slosh around. You will then take your paintbrush and dip it into the ethanol to sterilize it, then into the water to rinse off the ethanol. Using this damp brush, pick up some seeds out of the tube. You can drop them in places around the filter paper. You may need to move some around. There should be 2 seeds on each wedge. Cover up the plates and place them in the fridge at 4 degrees Celsius for 1 week. While you are waiting, randomize your plants using the reference numbers and fill the flats with soil. We have labeled ours with lab tape across the top and down one side. Our columns are numbered and our rows are lettered. Once the reference numbers are assigned random positions, you can sort by reference number and print out slips of paper that tell you where each pair of seeds in each Petri plate goes. You will also want to print out the list for the whole flat for proofreading later on. After a week in the fridge, you'll be ready to plant out the seeds. We suggest setting up a large operation in an empty room with several students. Here, we have 2 tables with our flats and 1 table that has all of the Petri plates and slips of paper for position assignments. A student will take a Petri plate and position assignment from the front table and distribute the wedges accordingly. When wet, these planting wedges are easy to break apart. Make sure the seeds have not shifted before you start distributing the wedges. Follow the position assignments and place each wedge from the plate onto the appropriate cell of the flat. Proceed until all wedges have been distributed. Before washing the seeds onto the soil, you'll want to proofread every flat. Take the printed whole flat list and make sure that the reference numbers on the wedges match the positions listed on the sheet. Once that is done, you are ready to put the seeds on the soil. Take a wash bottle of water and carefully rinse the wedge so that the seeds drop onto the soil surface. Check the wedge to make sure that the seeds are gone. Then you're done. Put on a germination dome and place the flats in a growth chamber.